Okay, so today we are going to start going over some mutations, and we're going to start off with chromosomal mutations first. So let me just explain to you guys, because we're going to go over two different types of mutations. The first one here, if we have a whole chromosome, the chromosomal mutations is something is going on with this chromosome. We have some kind of mutation that occurs in it. Now, later on, we're going to go over mutations in our DNA. So how is that different than a chromosomal mutation? Well, a chromosomal mutation is going to affect a large segment of our DNA. So let's say if this area right here that I'm circling, let's say this area is affected. Well, if you remember how we make a chromosome, we take our DNA and it gets wrapped around and condensed around all of our histones. So we have a whole lot of DNA spiraled, and that creates a chromosome. So if we're talking about this area right here, I'll use a different color there, but if we're talking about that area right there where there's a chromosomal mutation, we have a whole lot of DNA that has this mutation in it. Okay, so something happened to that very large segment of DNA in that area. That is a chromosomal mutation. Now, if we are going through a DNA mutation, it's just going to be one point on our chromosome. So it's just going to be one area right there, there's a mutation. So chromosomal mutations are much larger than just DNA mutations. Chromosomal mutations, we have a large mutation with a whole segment of DNA. With a DNA mutation, it's going to be more localized to one area, and there might just be one small error in any particular part of the DNA segment. All right. So mutations happen, guys. They happen all the time. We can have a DNA one. We can have a chromosome one. You guys just saw the difference there. Now, mutations are not always bad. Some people are like, oh my goodness, mutation, I'm going to get cancer. No, not always. Now, for us, we don't see mutations as prevalent as other organisms. Bacteria, viruses, which really aren't organisms because they're not living, they mutate all the time. Reason being is they reproduce very, very quickly. So while we might have one generation of humans, bacteria might have billions upon billions upon billions of generations occur because they're reproducing so quickly. So mutations for us aren't as prevalent as the bacterial ones. But regardless, what they can do, along with causing cancer, which does happen sometimes, is they can actually increase genetic variation. They could bring about new traits and new alleles in a population. So let's just go over the chromosomal mutations first. Our first one, we have a chromosome here on the right. First one is the deletion. So all we're doing is taking this segment right here, that area right there, right? And we are deleting it. So if we delete that area, all of those traits there, all of these traits, they're gone. So you will be missing some alleles. You will be missing some characteristics for certain traits. All right. So that is a deletion. Second one is an inversion. Now, inversions aren't a big deal. They will affect some traits, and they will affect crossing over later on. But if you notice, all we're doing is taking a segment of chromosomes, or a segment on a chromosome, and flipping it. That's all we're doing. We're flipping it upside down. All the traits are still going to be there. They are just going to be backward. And like I said, it's not a huge deal, but we do get some traits affected by it. And then when our cells go to reproduce to make sex cells, we will have some issues there, especially when it comes to crossing over. Because these are switched, we might get 3 of one allele or just 1 of other alleles. And we'll talk about, if we have more or less than one chromosome, what happens there later on. Next one is called translocation. So we got trans and we got location in there. So location, what happens is this piece of a chromosome actually changes its location. So it breaks off of one chromosome and goes to another. Now it's not going to be the same chromosome. If you remember, like we got chromosome 4 down here. You're going to get a chromosome from mom and a chromosome from dad for number 4. But what happens is that segment on chromosome 4 actually goes to a whole different chromosome. So those traits are being expressed on the wrong chromosome. This could be really bad, just because if they are expressed on a different chromosome, maybe a different type of cell, if it's a heart cell, a lung cell, it might be expressing something that it shouldn't express, or it might not be expressing something that it should. Okay, the next one, and this is the one I kind of touched on. It's called non-disjunction. So here is what happens. Non-disjunction is our chromosomal mutation that is very, very, very bad. And usually on the keystone, this is the one they like to put on, just because it is so serious. Now, in the past, we went over a condition called Trisomy 21, if you remember. Trisomy. And if we have tri, that means 3. So that tells us we have 3 of chromosome number 21. On the other hand, we can also have monosomy. And mono means 1. So this means having 1 of any chromosome, and then you'd put the number there. Now, most of the time, if it is a monosomy, that individual is not going to be born. There's going to be a miscarriage. The only one that there is a possible chance of them being born is monosomy X. It's on the sex chromosome. It's the only one where we've had individuals born that have been missing a full chromosome. Now, if you remember on our karyotype, we went number 1 through 23, and as we went higher in our number, our size of our chromosomes got smaller. So number 1 was our biggest chromosome, number 22 was our smallest chromosome, and then 23 was our sex chromosome. So we're not going to really mention those right now. But what happens in non-disjunction? If you remember, we were talking about the law of segregation during meiosis, where these, our chromosomes here, are going to separate to opposite poles of the cell. Now, if they don't separate, and I'll just draw you a picture here. Let's say we have, I'm just going to do 2 chromosomes here. And let's say our bottom one separates. So we get 1 over there, we get 1 over there. And then on the top, that one doesn't separate. It all gets pulled to one side. Well, when that cell forms, we're going to have the full X, the 2 chromosomes there, and that one. And the other cell, we're just going to have that. So this one is going to have 1 more chromosome than normal. This one over here is going to have 1 less chromosome than normal. This is non-disjunction. This is when the chromosomes, they fail to separate. So one cell, sex cell is going to have more than 1. One cell is going to have less than 1. Now, this does not run, especially like trisomy 21, Down syndrome. It doesn't really run in your family. It's not, it is a mutation, but it's not really genetic. What happens here is just the chromosomes really fail to separate. Something is going on where we had all those checkpoints. One of the proteins at the checkpoint is not working properly, and yeah, we're not separating, so not good. All right, so some mutations can be passed on. Some of them cannot. Some cancers can be passed on to the next generation. Others, maybe there's just a mistake going on in your body that causes you to get some form of cancer. So this person here has skin cancer. So always wear sunscreen, guys. If you can see here, obviously like his ear is just crazy. But he definitely has some kind of melanoma, all right, for the skin cancer. Not good. Smoking and chewing tobacco. This picture is from chewing tobacco here, where the guy has this gigantic thing on his tongue. When you do these types of things, there is a lot of carcinogens, a lot of bad things in the cigarettes and the chewing tobacco, and it does cause an increased prevalence of mutations in your body. Do those mutations lead to cancer? No, not always. But in the frequency of which you do things, if you're smoking more cigarettes, you're continuing to smoke more and more and more throughout the day, yeah, you have a higher risk of getting a mutation and a higher risk of developing cancer. Now, sometimes some people might just, smoke, they smoke maybe for a year and then they develop lung cancer. This could happen too. You don't have to be a chain smoker your whole life in order to develop lung cancer. So mutations can still come about. Okay, so mutations in sex cells. And we already talked about non-disjunction was the big one. And that's when we're going to have one of the parent sex cells giving more chromosomes than the 23 for humans. Okay. So, like I said, there's mutations. Some of them are good. Some of them are bad. So the cheetah here, they are very, very fast. Now, the cheetah actually, we predict the cheetah is actually going to be extinct many, many, many years from now, just because there's no genetic variation left in the cheetahs. There's very little. The cheetah actually is not evolving too much anymore. And in order for a species to continue to live, it has to continue to evolve, but we'll get into that a later chapter. But if a cheetah has a mutation to make it run faster than the other cheetahs, that's a really good mutation. If an individual has sickle cell anemia, we talked about that in the past, where the red blood cells are sickle-celled and they can't transport as much oxygen, that's a bad one. Hemophilia, the inability for your blood to clot. If you have that mutation, that's really bad, okay? So these types of diseases probably weren't in humans for a very long time, and then someone got a mutation for it. That person reproduced, and it became more prevalent, and so on and so forth. Okay, so we're going to start to go over DNA mutations. I'm only going to go over maybe one or two today, and then we'll call it a day. So we're done with the chromosome mutations. We're done with all of those. These are going to be more localized to one area. So let's start off with point mutations. So what's going on with point mutations is we have one nitrogen base somewhere in our DNA that has something wrong. Okay? Now, in order for you guys to fully understand these, make sure you have your codon charts. They will help you out to solve these problems. So if you remember, we had the circular one and then we had the box one. They'll really help out. So let's say we have AUG. Now, I'm just doing an RNA one here, and I'll show you the long way later on, but let's start off slow. So AUG on our charts goes to methionine, MET. Now, what I did was I mutated that segment. So we had a U here. So we had the U. And let's say that mutated to an A. If it mutates to an A, AAG now goes to a completely different amino acid compared to AUG. AUG goes to methionine, AAG goes to lysine. So that mutation caused a complete change in that amino acid. All right, so if you guys see here, here's just a picture of one. We took the T and it mutated to a C. Okay. All right, so the first one was called a missense. That was our, if I go back here, that was our missense mutation. We changed the amino acid that we were getting to. Our second one, this is still a point mutation, but it's called a nonsense mutation. So what happens here is we're transcribing and translating, everything is good, and then all of a sudden we have a change in that DNA sequence. And just like anything else, guys, we always want to go left to right, just like you're writing. And when we do this, so we have GLN, 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 and then all of a sudden the C mutates to a T. And when we put this in our chart for our codons, that leads to a stop codon. This is called a nonsense mutation. So in a nonsense mutation, we get a premature stop codon. When we have this stop codon, that's it. Everything stops. All of these other ones that are past it do not get translated, okay, because this is a termination signal. It literally stops everything else from continuing transcription and translation, all right? So that's it. Once we get a stop, you guys don't have to put anything after it, all right? Nothing else will go there. All right. Okay, guys, let's stop here for today, and we will start here. Next time, I'll go over the rest of the point mutations and then the more serious mutations that could really screw things up. All right, guys, have a good rest of your day, and I'll see you next time.